The Cell Cycle by Gabby and Lila.
Gap 1 is the start of the cell cycle. This is where the cell makes a decision to go into mitosis, and it makes sure it has the nutrients it needs to divide.
During this stage, a cell can enter gap 0. This stage is where a cell can no longer divide and stays in the state until it dies. In special circumstances, a cell can exit the state.
The second stage of interphase is called synthesis. During synthesis, the cell replicates its DNA, so each DNA has a duplicate of itself identically. During this stage, it's still hard to see the DNA since they are still in their chromatin form.
The next stage is gap 2. This is where cell grows and take in nutrients to prepare to divide. The cell stops to make sure that all the DNA was replicated correctly, and the centrioles are duplicated.
Gap 1, synthesis, and Gap 2 all make up interphase in the cell cycle.
The part of the cell that actually divides is mitosis. It's made up of prophase, metaphase, anaphase, telophase, and cytokinesis.
That is the cell cycle. During interphase, the cell prepares for mitosis and carries out its cell duties.
During prophase, the chromatin condenses into sister chromatids attached at the centromere. During this stage, the nuclear envelope dissolves.
During metaphase, the sister chromatids line up at the equator of the cell. During this stage, the spindle fibers attach to the sister chromatids.
During anaphase, the spindle fibers pull the sister chromatids apart, bringing one chromatid from each pair of the opposite sides of the cell.
During telophase, two new nuclear envelopes form and spindle fibers retract.
Cytokinesis, the cytoplasm divides and the cell officially divides into two daughter cells. In animal cells, the cytoplasm pinches inward, creating a cleavage furrow until it cinches the cytoplasm in half, dividing the cell. In plant cells, a cell plate forms in cytoplasm until it cuts the cytoplasm in half and creates a new cell wall.
And voila! Now there are two daughter cells.